Right, this video is going to sort of be an intro to linked genes, but we're going to actually go over unlinked genes, which is really what we've been talking about up until this point. So, what you need to know is that genes can be linked. What that means is when genes are linked, they are located on the exact same chromosome. And some chromosomes have thousands of genes, so all the genes located on a single chromosome actually together act like linked genes. But before we get into that, let's, for comparison, talk about what we just sort of visualizing on chromosomes, what we were always talking about before. We've been imagining genes to be like this. I have homologous pairs of chromosomes, and I have a capital A, lowercase a, capital B, lowercase b. That means the genotype of this individual is this. And based on independent assortment, that would mean that the gametes that this person could form based on this genotype, right, again, because of foiling and whatnot, these are the possible gametes that they would be able to form. And we can do a test cross, and a test cross is where we will cross with a homozygous recessive individual. So if we cross with a homozygous recessive individual, this is the Punnett square that we would be using. This is obviously our individual that we're interested in. This is our homozygous recessive individual. And the offspring that we can have are in the darkened boxes here. We can get 25% of 4 different categories. And so say, for example, out of 400 offspring, we would get 100 of each of these. That is how we've been talking about unlinked genes. In the next video, I'm going to show you how linked genes are different than this. So in this video, I'm going to be talking about the basics of linked genes. So unlike unlinked genes, which I showed you about in the last video, linked genes would instead look like this. We have 2, again, homologous chromosomes. Notice again that the A gene is located in the same place on both chromosomes, and same with the B gene, located in the same part of the chromosome as the other B gene. But notice we have genes literally linked together. They are attached to each other. In the way that I happen to draw it here, the capital A and the capital B happened to be attached to the same chromosome, and the lowercase a and the lowercase b happen to be attached to the same chromosome. So again, our individual's genotype is the same as what it was before. So the genotype has not changed. The phenotype has not changed. This is all about how the sort of inheritance of this trait through their offspring is going to be changed. So when the genes are linked, they can only be inherited in one type of combination. So even though this is their genotype, we can no longer do foiling because there is no independent assortment. They literally assort together. So we can only get capital A, capital B, and lowercase a, lowercase b, because those are the 2 chromosomes that will separate during meiosis. Therefore, with only those 2 options for gametes, we can again do a test cross, and this is our Punnett square because we only have 2 gametes this time. We now have 50% of each possible offspring. Notice we now only have 2 options for offspring. And out of a same 400 total, we now have 200 of each. Notice also that the phenotypes of the offspring we're getting are the exact same as our parents, and we call these the parental phenotypes. They look exactly like the parents. So this is the basics of what the difference is with linked genes. You would look at a cross, might look at this cross and say, wow, I can get, if I foil, do a whole Punnett square, I get 4 possible offspring. But when I actually do this cross, I actually get 2 offspring and they look exactly like the parents. That is a sign of these genes being linked. And also will tell you what the linked combinations were.